It's a pleasure to be here today. And I guess to give a brief little context, the way this talk may differ from some of the other ones today is that while a lot of the talks have focused on prediction, how can we predict an outcome, in this talk and a lot of genetic epi work, we don't really care too much, often care about predicting disease, but really discovering what may be associated with disease. So I'm going to talk a lot about the right-hand side of the equation as opposed to the left-hand side. So a brief outline, I'm going to give some background and motivation, and I hope a lot of you are here for Marsha Wilcox's talk because she gave a much better in-depth overview of genetics than I possibly could. And then I'll walk through an application of RandomForests to a data set. Then I'll follow it up with a simulation study to examine some of the results and conclude with just some discussion of future work. So in genome-wide association studies, we study a unit of DNA referred to as single nucleotide polymorphisms, or SNPs. And there's millions of these SNPs across the genome, accounting for much of human genetic variation. And each SNP consists of a pair of biallelic alleles. And as a unit of data, we can think of these as a pair of 0-1 variables. And if with the probability of observing a 1 referred to as the minor allele frequency, or the MAF. So if we have, for any SNP, we can have 3 realizations within the data. We can have, if we code the SNP as big A and little a, with big A referring to the major allele, we can observe 2 big A's, which would be a 0-0, a big A and little a is 0-1, or 2 little a's, a 1-1. When a SNP has some impact on some phenotypic variation, there's generally 3 different types of causal mechanisms that can occur. We can have an additive effect where each additional presence of the minor allele causes the variation, essentially as a dose response. We can have a situation where any presence of a minor allele causes variation, a 0 versus a 1-2 situation. Or we can have what's referred to as a recessive effect, where one needs 2 copies of the minor allele to have the observed variation. Then in genome-wide association studies, they're a very typical, simple case control epidemiological study design. You recruit thousands of people with and without a particular disease of interest. You type each person on nowadays up to 1 million SNPs. A few years ago, this would be a couple 100,000, but as genotyping has gotten cheaper and more efficient, this number has grown. And the goal is simply to determine if there is differences in the frequency of a given SNP between those with and without disease. This is generally done by using a chi-square test with then some degree of control for multiple testing. And the results, and the standard today are for the results to be confirmed via replication in an independent data set. Of course, as presented, there's lots of complications. SNPs, as a unit of data, will exhibit a local correlation structure referred to as linkage disequilibrium, or LD. Essentially, 2 SNPs that are located near each other on a given chromosome will exhibit this correlation, while SNPs that are farther apart or on altogether different chromosomes will not be correlated with each other. There's likely a high degree of unknown heterogeneity among different disease subtypes, and it's really important to note that SNPs are just one component of genetic variability, and while a disease may have a genetic component, that may or may not be expressed among these SNPs. From an analytic perspective, there's a relatively high noise-to-signal ratio, where even though we're looking at hundreds of thousands of SNPs across tens of thousands of genes, we don't presume that all of these are involved in the disease process. Instead, maybe only a few dozen or up to genes may be associated with disease. So really, we think of a GWAS as really a scan across the genome trying to find regions of interest, essentially a large needle in the haystack problem. That being said, it's not like we're looking for a magic silver bullet that has a large effect, but instead disease is likely caused by many different weak effects spread out across the genome, again, making analysis difficult. And just to note, the causal mechanism is often unknown and will vary from SNP to SNP. This then obviously presents a lot of problems. While the allelic chi-square test is a fairly robust test to all alternatives, since it does not take into account any causal mechanisms, also not always a very powerful test. And when we add into account, as was talked about just beforehand, if interactions or high-order effects, we're going to have even less power. And while in theory, in principle, we can look at all different types of causal models, all different interactions, this is computationally challenging and just subject to enormous degree of multiple testing. So the growing standard in the field of genetics is it's becoming more of this using machine learning algorithms as maybe an approach to allow one to consider a greater number of effects. So what are really machine learning algorithms? And this is something that we've been talking about throughout the day today and tomorrow. I think of the machine learning approach is really to search over some partition of the full multidimensional space, and it's this partitioning that allows for the consideration of these joint and conditional effects. Now, each algorithm is going to partition the space in a slightly different way, so that when we get back rankings of variable importance, we should think of these variables as important and interesting to the given algorithm and not necessarily a hypothesis test of causality. We wouldn't expect two different algorithms that are partitioning the space differently to come up with the exact same ranking of interesting SNPs. These are really just markers that are important to the given algorithm. There's a lot of different algorithms that are now being used in the genetic literature, some which have been developed specifically with SNP data in mind, and then others which are more general algorithms that are now being applied to genetic data. So what I'm going to talk about is the random forest algorithm. It was written about in 2001 by Leo Breiman, and it's really kind of a logical culmination of two of the seminal works. One was on CART with, that he did in the mid-'80s with Richard Olshan, Jerome Friedman, Chuck Stone, and then his work in the mid-'90s on bootstrap aggregation or bagging. So like in bagging, one starts by selecting a bootstrap sample of the data. Then you select a random subset of all the variables in your data set over which to search. Then as in CART, you find the variable that best splits the data into homogeneous groups. This entire process is repeated over and over again until a CART tree is formed, though one of the key distinctions between CART is that you don't go about pruning the tree, you keep the tree as large as possible. Then after the tree is formed, you run the data that's not in the bootstrap sample, referred to as the out-of-bag data, down the tree, and this is how you get measures of variable importance and error rate, the error referred to as the out-of-bag error rate. This is then repeated over and over again until a full forest of trees is grown. So it's fairly well understood why this works well as a predictor. As most of us are aware, we can break down prediction error into the two components of bias and variance. So CART trees are known to be generally low bias classifiers, though highly variable. Essentially, any perturbation of the data will result in a slightly different classifier. Bagging as a process is able to reduce the variance of the base learner while maintaining the initial bias of that learner. However, in bagging, there's a lower bound on how much variance reduction we can get, and that lower bound is essentially the correlation between the individual trees. By choosing a random subset of the data at each node, we're able to reduce the correlation between trees, further reducing the variance of the overall classifier. In terms of how it works for variable importance, this is actually where the more interesting work is going on. But one of the things that is known is that unlike a lot of other algorithms, such as the last few or boosting algorithms, each variable is searched over, so has a chance to be put into the model, leading ultimately to a much less sparse solution, something more akin to a ridge regression. It's not clear if this is a good thing or a bad thing, it's just something important to acknowledge. Though it's really the CART-based tree structure that gives it its strength and allows for all these conditional effects to be considered. Now, in dealing with classification, there's two main types of variable importance that are used. One, the more general one, is permutation importance, where one simply permutes the labels of the out-of-bag data before sending them down the tree. So any increase in misclassification after this permutation, it becomes the error rate, becomes the variable importance for that variable. And then the Gini importance, where, so the Gini criteria is the criterion used in constructing the CART tree. It's similar to a mutual information or criteria, and the decrease at Gini at each node becomes the measure of variable importance. So in terms of some uses and advantages, it can be used, like many algorithms, to get rankings of important variables. The forest can be used to predict new observations, and it can be used, and the dense forest of trees can be used to detect clusters of observations and predictors. One of the key advantages is this internal form of cross-validation via the out-of-bag error rate, which does save a lot on computational time. It works well with both continuous and categorical outcomes and predictors, as is the nature of CART. From a genetic perspective, it can accommodate very large data sets, and actually, since all the trees are grown independently, it's very easy to make embarrassingly parallel. And it's thought, for the most part, to be fairly robust to different tuning parameters, making it a fairly easy-to-use off-the-shelf method. So at this point, what I would say is known is that it's a very strong classifier with more work being done on the variable importance measures. And for the most part, most applications have been run fairly successfully with just default settings.